All right, let's jump into this deep dive. We've got a really fascinating stack of sources today, basically pulling back the curtain on how life is built and how it works, from the tiniest scale up.
Exactly. We're talking about, how cells stick together, the mechanics of tissues, how cells behave, things like merging together or moving around.
Yeah, and then how we model all that using different kinds of networks, imaging techniques, plus some uh, really big ideas about evolution.
And even touching on something maybe less talked about, bioelectricity's role in it all. It's quite a range.
It is. So our mission here is to sort of unpack this, pull out the key insights, and help you connect the dots across this, well, pretty complex landscape of biology. Find those aha moments.
Sounds good.
Okay, let's zoom way in. Start right at the edge of the cells where they meet. Cell junctions. The sources spend a good bit of time here.
They make a key distinction right off the bat. You've got bicellular junctions, where just two cells touch.
Okay, simple enough.
And then tricellular junctions, or TCJs, that's where three or sometimes even more cells all converge at one point.
Ah, okay, like a corner.
Pretty much. One source uses the term vertices for these tricellular meeting points. It's a useful geometric term.
Vertices, got it.
And the important thing is these aren't just, passive corners. They often have specialized structures. Looking closely with microscopes or specific molecules, you see things like tricellular tight junctions or tricellular septate junctions.
Just specific machinery right at that three-way point to seal things up.
Exactly. Really important for barrier function, like in epithelial tissues. Though, interestingly, the sources mention you also see tricellular contacts in mesenchymal cells, looser connective tissue cells.
Oh, but are they the same?
Apparently not quite. They seem to lack those really distinct, specialized junctional structures you see in epithelia. So the contact is there, but maybe less complexly structured.
Interesting difference. Now, the sources also mention something about adherence junctions and tight junctions being kind of mixed up sometimes.
Yeah, they're not always perfectly segregated into neat lines. And there's this really fascinating observation linked to that. Adherence junctions, the ones providing strong adhesion, they actually dissolve when a mammalian embryo hatches from its protective layer, the zona pellucida.
Huh. Why does that happen?
Well, the source connects this to cancer metastasis. The idea is maybe getting rid of these strong cell-to-cell grips is a crucial step that lets cancer cells break free and move.
Wow. So the embryo dissolving adhesions to emerge is kind of like a model for cancer cells breaking loose.
It's a striking parallel they draw. The embryo doesn't need to squeeze through a tight spot once those adhesions are gone, and maybe cancer cells exploit a similar mechanism to escape a tumor.
That really highlights how developmental processes can be, well, repurposed in disease. And speaking of barriers, epithelial sheets, like our skin or gut, lining that barrier function against pathogens is absolutely vital, right?
Yeah.
It needs to be strong.
Absolutely critical. And the sources dive into the molecules that make it strong, especially at those tricellular junctions, which could be weak points. They highlight a protein called tricellin.
Tricellin. Okay, what's its job?
Its main role is to really secure that epithelial barrier right at the tricellular junction. It acts like a specialized anchor there.
An anchor for what? How does it work?
It links the junction itself to the cell's internal skeleton, the cytoskeleton. Specifically, tricellin anchors F-acting part of the cytoskeleton via another protein called alpha-catenin, which is a key scaffold for adherence junctions.
Okay, so it's connecting the sealing parts to the structural and force-generating parts of the cell.
Exactly. It couples the seal with the cell's ability to contract using ectomyosin. This whole connection is vital for making those tricellular tight junctions work properly.
And it's not instant.
No, the sources mention that forming a distinct actin meshwork right at the tricellular junction is like a specific step in the junction maturing and getting strong.
Fascinating molecular detail. But it's not just molecules. The sources also talk about the physics of these epithelial sheets.
Right. They point out that the sheet stiffness, its rigidity, isn't constant. It can be tuned, changed by things like mechanical strain or just the overall shape of the cells. Are they tall columns, cubes, or flat squamous cells?
And modeling helps understand this.
Yeah. Simple 2D models often just show tissues getting stiffer when you compress them, but the sources highlight that models considering the third dimension, the cell height.
Ah, the actual 3D shape.
Right. These models predict that the tissue can get stiffer not just when compressed, but also when it's stretched or dilated away from that sort of neutral cuboidal state. It shows how fundamental the 3D geometry is to the mechanics.
That makes sense. Okay, so we've got cells building structures, linking up, having specific physical properties. But they're not just static bricks, are they? They move, they interact. Sometimes they even merge.
Absolutely. The sources bring up cell fusion, leading to syncytia.
Syncytia, right? Those are single giant cells with multiple nuclei inside.
Exactly. Formed when individual cells fuse together. Muscle cells are the classic example most people know.
Okay. And the sources mention other examples.
Yeah, particularly in the worm C. elegans, which is a workhorse for developmental biology. They list a worm's skin, hypodermis, parts of its reproductive system, vulva, uterus, an excretory gland, even in how gene cells form and certain throat muscles. Fusion happens quite a bit there.
And why is this fusion event so significant for, say, tracking development?
Well, think about how we usually draw cell lineage, like a branching tree, right? One cell divides into two, they divide, and so on.
Yeah, binary tree.
Cell fusion breaks that completely. When two or more cells fuse into one, your nice, neat tree structure suddenly becomes a more complex network, a graph. The branches merge back together.
Ah, I see. It messes up the simple tree diagram.
Totally. And it's not rare. The sources state there are 44 known cases of somatic cell fusion just in C. elegans. That's 44 points where the lineage stops being a simple tree.
44. Wow, okay, that really changes the picture of development.
Yeah.
And besides fusion, cells also just move, right? Migrate.
Definitely. During development, cells migrate from where they're born to where they need to end up in the adult body. And they often retain some plasticity, some ability to change or move, even after embryonic stages, but before the animal is fully adult.
How do researchers track that movement?
The sources mention a couple of techniques. One is particle tracking velocimetry, or PTV. Basically, you track tiny particles, or the cells themselves, and calculate their velocity, speed, and direction.
So measuring how fast and where they're going.
Yep. And another concept is Lagrangian coherent structures, LCS. It's a more mathematical way to analyze the flow within the tissue, identifying sort of highways and barriers that guide or impede cell movement, like finding the hidden currents the cells are following.
Okay, so tracking individuals and analyzing the overall flow patterns. Now, trying to understand all this complexity, the structures, the fusion, the movement, often means we need abstract ways to represent it, right? Like using graphs.
Exactly. And the sources introduce an extension of graphs called hypergraphs.
Hypergraphs. How are they different?
So a normal graph edge connects just two nodes, like two cells. A hyperedge can connect three or more nodes all at the same time.
Oh, okay. So it can represent group interaction.
Precisely. It's much better for things like that tricellular junction where three cells meet, or for representing a cell fusion event where multiple cells merge into one. It captures relationships beyond simple pairs.
This seems really useful for development.
It is. They also mention hypernodes, which can represent categories or groups of nodes or even internal features of a cell. Using hypergraphs for something like the C. elegans lineage makes a lot of sense because of all that fusion.
And does this help with computational analysis?
Yes, the sources note that if you add positional and structural information into these graph or hypergraph representations, it can significantly boost the performance of machine learning tools, like graph neural networks, when analyzing them.
Basically, giving the algorithm more context makes it smarter.
Kind of, yeah. You can even use standard network measures like degree distribution, how many connections a node has, but apply them in the context of hyperedges.
Got it. And beyond lineage, this network thinking applies to tissues too.
Yes, translating a whole tissue into a spatial network, cells or nodes, connections or edges, allows for mathematical analysis of its organization.
And then there are the ultimate biological networks, connectomes, the brain's wiring diagram.
Right, networks of neurons, synapses, gap junctions. And here the sources highlight something really intriguing, a recurring pattern called the XRR motif.
XOR, like the logical operation.
Kind of, yeah. They found this specific little circuit pattern repeated in the connectomes of very different animals, C. elegans, the fruit fly Drosophila, even in the mouse visual cortex.
Whoa, across different species. What might it be doing?
The suggestion is it could be a fundamental computational unit. Maybe it helps balance excitation and inhibition in the network, maintain stability, homeostasis.
Okay, that makes sense.
But they also propose, maybe more speculatively, that it could act like a basic loss function for learning or plasticity, a simple circuit that helps the network adjust its connections based on activity.
A built-in learning rule.
Potentially. Though they do caution that its specific role in driving plasticity really needs more confirmation from models that simulate network activity over time. Still, finding a common motif like that is pretty exciting.
Absolutely. A conserved circuit, possibly linked to learning. Now, zooming out again, The sources also think about how tissues and organs might evolve in the first place. It's not just about getting new functions.
Right. One idea is that it might be more about integrating functions that individual cells already had, but organizing them into coordinated functional networks.
And what drives that integration?
A key idea proposed is offloading metabolic burdens. Basically, specialization and division of labor. Instead of every cell doing everything, some cells specialize and support others.
Like in an economy.
Kind of. The examples given are great. Astrocytes in the brain store energy, glycogen, and handle neurotransmitter cleanup, glutamate, taking the load off neurons so they can focus on firing signals.
Okay, yep.
Or another example, pancreas acinar cells, which secrete digestive enzymes. When they become part of the structured pancreas tissue, they seem to become dependent on metabolic support from other cells in a way their evolutionary precursors might not have been.
So integrating into a tissue allows for, or maybe even requires, this metabolic cooperation and specialization.
Seems to be a powerful driver. It's an efficiency gain, managing complex metabolic tasks better as a team.
Okay, shifting gears from structure to simulating behavior, the sources mention some pretty complex models.
Yeah, things like brain-body environment models. They use the AI worm model for C. elegans as an example.
Brain-body environment, what does that involve?
It's quite holistic. You combine a model of the worm's neural network with a biomechanical model of its physical body, often built from like interconnected virtual springs or tetrahedrons representing muscles and tissue.
So a virtual worm body.
Exactly. And then you place this whole simulated organism into a simulated physical environment, maybe with food gradients or obstacles. This lets you study how neural activity translates into muscle contraction, which then produces movement within that environment.
Simulating the whole loop from brain to behavior. Cool. And how do they make the movements look realistic?
They mention using techniques like inverse kinematics. Instead of simulating the muscles first to see what movement results, you define the movement you want, say, the sequence of body shapes for slithering like a snake.
Okay.
And the inverse kinematics algorithm works backward to figure out the necessary joint angles or muscle activations needed to produce that specific trajectory. It's a good way to generate biologically plausible motion for simulations.
Clever. Working backwards from the desired outcome. And linking models to real data, the sources touch on imaging, specifically CT scanning.
Briefly, yeah. Computer tomography, they mention different setups like parallel beam versus fan beam geometry for collecting the data. A key challenge for biological tissues, compared to, say, scanning a bone, is light scattering and noise within the tissue itself.
Right. Tissues aren't perfectly transparent or uniform.
Exactly, which makes reconstructing a clear 3D image tricky. And they use this brilliant analogy. They compare the iterative reconstruction process, building the image piece by piece, from noisy data to playing Wordle.
Wordle, how so?
You make an initial guess, start reconstructing, you get feedback on what's right or wrong, how well it matches the messy data, and you use that feedback to refine your next guess, getting closer and closer to the actual image, just like you zero in on the word in Wordle.
Huh. That's a fantastic analogy. Makes a complex algorithm instantly intuitive.
It really does.
Okay, so we've gone from cell connections, movement, modeling. Let's take that step back now to the really big picture. Evolution. The sources have some major points here, starting with the origin of eukaryotes.
Right. The emergence of eukaryotic cells with a nucleus and organelles is seen as a massive turning point in life's history. It really paved the way for complex multicellularity.
And one source calls it an algorithmic phase transition. That sounds profound.
It does. The idea is that it wasn't just adding parts, but a fundamental change in the rules or the algorithm by which the cell operated, managed information, and handled energy.
How does that connect to organelles?
It ties into the endosymbiotic theory, the idea that organelles like mitochondria were once free-living bacteria that got incorporated into a host cell. This brought new functions, but also demanded a whole new level of internal organization and genetic coordination.
Which links to the genome differences between prokaryotes and eukaryotes.
Exactly. Prokaryotes, like bacteria, usually have small, tightly packed genomes, very efficient, not much non-coding DNA. Eukaryotes have these much larger genomes, loads of regulatory regions, introns, non-coding stuff.
And why is that important?
The argument in the sources is that the prokaryotic genome structure might have limited the size and complexity of proteins they could easily make, putting a cap on overall cellular complexity. Eukaryotes, with their different gene structure and regulation, overcame that bottleneck.
So the eukaryotic genome structure itself was a key innovation, allowing for more complexity.
That's the idea. It unlocked new possibilities. Then there's this other powerful metaphor one source uses: life as a cascade of machines making machines.
Machines making machine.
Yeah, thinking of biological processes from molecules up as hierarchies of machines that build and replicate each other. Self-reproduction is the key defining feature across all these levels.
And these aren't just random processes.
No, that's the point. Even things like ribosomes or molecular motors operate with what the source calls a non-random bias. They have enough precision and consistency to perform specific tasks reliably, unlike just random thermal noise.
Okay, and how does this relate to survival?
Survival, in this view, is fundamentally about physical continuity, just persisting through space and time. And living systems, these cascades of machines, are exceptionally good at finding and maintaining these trajectories of continuity compared to non-living things.
Because the ones that weren't good at it
Yeah.
just aren't around anymore.
Essentially, yes. We only observe the systems that have successfully navigated this process of persistence over vast timescales. It's a form of selection based purely on continuity.
And they contrast this with, say, a rock.
Right. A rock persists through sheer physical robustness. A river persists through slow change. Life's strategy is different. It persists through this constant dynamic process of self-replication, an infinite cascade of machines making more machines. Two very different solutions to the same problem of continuity.
Fascinating perspective. The sources also come back to the idea of trade-offs in evolution.
Yeah, especially metabolic trade-offs. Organisms are always juggling competing demands. The classic example is energy production versus damage control. Making lots of energy often produces harmful byproducts like reactive oxygen species, ROS.
Maximize ATP, but don't fry yourself with free radicals.
Exactly. And this constant balancing act is proposed as another reason why cells might have organized into tissues and organs.
How so?
Because forming an integrated tissue allows for specialization to manage these trade-offs more effectively. Some cells might focus on high energy production, while others specialize in detoxification or managing waste products, benefiting the whole unit. It's that division of labor idea again.
Makes sense. Distributing the risks and benefits.
And related to metabolism and size, they mention an optimization curve for microbes. It looks like there's a sweet spot for cell size, maybe around 1 micron, where the physics of how fast they can make ribosomes basically sets a limit on how quickly they can divide.
So fundamental limits based on the machinery inside.
Yeah, showing how molecular constraints bubble up to affect organism-level traits like growth rate.
Okay, we've covered a huge range: junctions, physics, evolution, metabolism. Let's turn to this last major theme: bioelectricity.
Right. And the key point the sources emphasize is that electrical potentials across membranes aren't exclusive to nerves. Almost all cells have them: muscle cells, stem cells, regular somatic cells. The signals might be different, maybe slower or steadier than nerve impulses, but the potential is there.
And this isn't a totally new idea in development.
No, there's a long, if sometimes overlooked, history of studying how bioelectric fields influence morphogenesis, how tissues get their shape during development.
But one source pushes this further, talking about an electrostatic force field. What's new there?
The novelty is thinking of it less as just localized potentials on individual cells and more as a continuous pervasive field existing throughout the tissue.
Like an invisible electric landscape the cells sit in.
Kind of, yeah. They propose this field could act a bit like chemical morphogens, the signaling molecules that pattern tissues, but using electrical forces instead. It could influence cell behavior and help organize patterns of membrane voltage across whole populations of cells.
So a physical field guiding development alongside chemical signals.
That's the provocative idea. It offers a different perspective, emphasizing continuous physical forces over discrete, localized computations or signals. They even propose ways to measure its influence, using terms like causal strength and causal distance.
Trying to quantify how far and how strongly this field might exert effects.
Exactly, and it raises big questions. Could this field allow for really long-distance communication between cells, even if the forces are subtle? Could it act as a sort of pre-pattern, setting up a basic template that other signals then refine and amplify?
It's definitely another layer of potential control and communication we might be missing.
It is. It challenges us to think beyond just molecules diffusing or cells touching.
Wow. Okay, this deep dive really has taken us on quite a ride.
Wow.
From, the molecular glue of tricellin and the weirdness of cell fusion.
Through to abstract hypergraphs, recurring brain motifs like XOR, thinking about life as machines making machines.
And these grand evolutionary transitions, metabolic trade-offs, and now this pervasive bioelectric field idea. It's a lot to connect.
It absolutely is, but it really drives home that understanding life requires looking at all these levels: molecular, cellular, tissue, organism, evolution, and using all the tools we have, from microscopes to math to physics.
Yeah, things that seem totally unrelated, like tissue physics and brain circuits, suddenly seem like parts of the same underlying puzzle of how living systems organize and persist.
The interconnectedness is definitely a key theme.
It really is. So building on that, maybe a final thought for you, the listener, to kind of chew on as we wrap up.
Mm.
As we get better and better at measuring and modeling biology in all these diverse ways, tracking single cells, mapping electric fields, using hypergraphs, seeing life as replicating machines, how does integrating all these different viewpoints actually change our fundamental grasp of what makes a living system tick? What does it even mean for something to be alive or functioning when we look through all these different lenses at once? And maybe more importantly, where might the next big breakthrough, the next key to unlocking life's complexity come from? What's the next unexpected perspective?